Hello grade 8s and welcome to the answer series Natural Sciences videos based on our study guides. This video looks at a practical experiment that allows you to test for the presence of starch in plants. The investigation must be conducted on a green leaf to ensure that chlorophyll is present. Geranium or malva leaves are especially great for this experiment because they are naturally soft and easy to work with. Start by placing the plant or a leafy stem of the plant in a dark cupboard for 48 hours. That's equivalent to 2 days. This will prevent the plant from photosynthesizing as there is no light available. In the time, the plant is going to use up all the starch that it's stored in its leaves to keep its metabolic activities going. The destarched plant is going to act as our control so that we can compare our results with it. To test for the presence of starch, you will need a green leaf, glass beaker, tongs, alcohol, iodine solution, water, a Bunsen burner or any gas hob, a test tube, and a white tile or petri dish. If you don't have any fancy lab equipment, don't worry, because you can still perform this investigation with everyday household items, using a small pot instead of a beaker, or a glass jar instead of a test tube. For the destarched leaf, I will be using the everyday household items and conduct the experiment using my stove because I don't have a Bunsen burner. The process may take a little bit longer this way and require the use of a bit more alcohol compared to when you're just using the test tube, but it works just as well. Then for the leaf that will have starch in it, I'll show you this same method using lab equipment. Take the green leaf and place it in boiling water for at least 2 minutes. You can increase this time because malva leaves are quite thick and sturdy, and the longer you boil it, the more sure you are that the cells are going to be killed and softened effectively by breaking the cell walls. Remember that cell walls are strong protective barriers around the plant cell, and we need to break these down to make the cell more permeable to the chemicals that we will be adding, like the alcohol and the iodine. Turn off the Bunsen burner or gas hob and use your tongs to remove the leaf from the boiling water and then place it in the glass jar or a test tube with enough alcohol to cover the leaf. Place the glass jar or test tube back into your pot or beaker with your boiling hot water. We call this setup a water bath. It allows us to heat the alcohol indirectly and not directly over an open flame because alcohol is highly flammable. So you need to be very careful when working with the alcohol and ensure that it doesn't boil. The alcohol is going to warm slowly and turn green as it extracts the chlorophyll from the leaf, turning the leaf white. A white leaf allows us to see any color change in the indicator more clearly. If the leaf was still green when we added the iodine indicator, we wouldn't be able to say if any color change indicated the starch being present or if it just reacted with the chlorophyll. So we want a nice white leaf to see a clear color change. You may need to heat the water in the pot or beaker again a few times just to keep the alcohol warm, but be careful not to add too much heat, so just intermittently add some heat so that the water starts to warm up and keeps this reaction of chlorophyll extraction going. You may also need to top up the alcohol, especially if you're using a glass jar instead of a test tube, because the alcohol does evaporate more than it would in the test tube. And there you have your white leaf. Place this white leaf back into the warm water for a few seconds to rinse off any excess alcohol and to soften it again. Lastly, spread the leaf in your petri dish and add a few drops of iodine solution to it. I've placed my petri dish on a white paper so that I can see a clear color change. Now this leaf was obviously destarched because it was in the dark cupboard for at least 2 days, so it shows no signs of starch. The iodine solution remains a light brown color, and this makes sense because there was no light for the plant to use for at least 48 hours. So it couldn't photosynthesize, it couldn't produce glucose, and no glucose was therefore stored as starch. Now you're going to take a different plant, or preferably the same leafy twig and plant because that makes it a more valid investigation, and you're going to place it in sunlight for 12 hours and repeat the investigation to test if any of those leaves now have starch present. So I'll quickly show you how to run through the same procedure using the lab equipment for this starch test. Once the iodine is added to the leaf, you'll notice a color change in the indicator. The iodine solution changes from a light brown to a very beautiful blue-black color, and it's a change that happens gradually as I add the iodine. So this is most assuredly a positive test for starch. So from this investigation, we can conclude two things. Firstly, that starch is present in the leaf because photosynthesis occurred. And secondly, that photosynthesis requires light, because starch was only produced in the leaf that was exposed to sunlight. No starch was present in the leaf that had been destarched in the dark cupboard. Check out the video description below for practice questions from our study guides. If you found this video useful, give it a like and subscribe to our channel so you don't miss any new episodes. Follow us on Instagram or Facebook to stay on top of the latest TAS news and launches. So that's it for now from the Answer Series, your key to exam success.